Good morning, everyone, and welcome to day 2 of GYSS. I hope you're all ready for another full day of stimulating plenaries and discussions. I'm Ian from the National Research Foundation, and I'll be your MC for today. Before we begin, a gentle reminder to put your mobile phones to silent so that we can all enjoy the program uninterrupted. As usual, there will be a Q&A session at the end of each lecture. We would like to remind on-site and virtual participants to submit your questions via the Slido platform. It can be accessed by scanning the QR code, and this can be done anytime during the sessions. Also, do follow us on social media for updates on GYSS. Search for gyss.sg on Facebook and gyss_SG on Instagram and Twitter. Join in the action by using the hashtags that you see on screen. All right then. Let's begin day 2 of GYSS. The first segment today is the Young Scientist presentations. We would like to invite Prof Langer of Duke NUS Medical School to chair the session, and our two presenters, Ms. Li Han-Chi and Dr. Patrick Fuchs. Prof Lok, please.
So good morning, everyone. My name is Shimei Lok. I'm the Provost Chair Professor from the Duke NUS Medical School. So the first speaker for this morning is Li Han-Chi. She's a PhD student at the Cambridge Institute of Medical Research. Her research interests include virology and host pathogen interactions. Han-Chi?
Morning, everyone. I'm Han-Chi, a PhD student in Professor Mike Weeks' lab at Cambridge Institute for Medical Research. And many thanks for having this opportunity to talk about my research on human cytomegalovirus. Thank you. So what is human cytomegalovirus? Well, hear this word, you might think, cyto, something sounds like a cell, and megalo, something sounds like a very big or giant thing. Then you are right. Human cytomegalovirus is a very big and complex double-stranded DNA virus with 170 genes. And of course, from its name, it's from the human cells. In fact, the human cells. So here's a picture showing the cells infected by the HCMV. And we can see that this virus occupies about half of the cells here, and also it can be spread between the cells very quickly. Actually, there are 60% of all of us in this room has been infected by the HCMV. However, it's normally kept silent in our bodies, don't worry about that. However, in the immunocompromised people, it can cause a substantial morbidity and mortality. So it's like a very high infection in the immunocompromised people, such as the people with the AIDS or the organ transplant recipients. Only a few people realize that the HCMV can affect 1 in 100 pregnancies and can cause the hearing loss and intellectual disabilities in newborns. However, there are only 4 anti-CMV drugs available, and all of them have significant toxicities and problems with drug resistance, and also there's presently no vaccine available for HCMV. So how this virus be so successful in defeating our cells? Actually, HCMV is a master regulator of our cell's defensive system. And one particular strategy it uses is to destroy certain cellular proteins. And the proteins are the cellular components responsible for almost every function in the cells. However, there are over 130 host proteins can be degraded or destroyed by the HCMV during the whole course of infection. And this is generally via the interaction of a viral factor shown here and its host target, followed by the recruitment of the waste dumps to remove the damaged part. And inhibiting this kind of interaction using the small molecules may form the basis of the new antiviral therapies. So the question is, how could we prioritize which cellular and viral proteins to study in more detail to give us this kind of antiviral therapies? We have recently established that the host factors of key functional importance can be destroyed during the early HCMV infection. And how could we measure this? The Weeks lab previously developed novel global approaches to identify individual proteins degraded to the fastest and also to the greatest extent during the HCMV infection. And this is all based on a technique called the proteomics, which enables more than half of all cellular proteins from different conditions can be measured in the same experiment. And these conditions normally include adding the waste dump inhibitors to rescue the protein from destruction, or adding the metabolic pause to measure the rate of the protein destruction during HCMV infection. And also, at the same time, the RNA, the transcript of the protein, can be harvested together and be quantified using the transcriptomics. And here's some data of a very good hit called the HLTF, which our lab has shown it to inhibit the HCMV's replication, so the virus needs to get rid of it. This protein can be rescued in the HCMV infection by the waste dump inhibitor shown here, and also it has a faster rate of destruction during HCMV infection compared to the uninfected cells, shown as smoke like here. And comparing the abundance of this protein and its corresponding RNA transcript, we found that the protein was decreased, but the RNA was not. And this indicates that it is the HCMV to destroy this protein instead of regulating its generation. So overall, our lab has shortlisted 35 proteins that destroyed by HCMV during early infection to the fastest and also to the greatest extent. And in addition to HLTF and another factor that our lab recently published, one very interesting hit that has been unexplored so far is DMX L1. So what is DMX L1? Actually, it's a very large protein formed by 3,000 amino acids. And also, it interacts with and stimulates the VTPase, which pumps the protons. So the stimulation of VTPase by the DMX-L1 can lead to an acidification of the intracellular membrane bond compartment in the cells, which is called vesicles, in particular, the lysosomes, which will be very important for the functional acid I will talk about later. So now, we want to address how and why does HCMV target DMX-L1 for destruction. So first question, which viral factors are involved and which host machinery is required? To understand which viral factors are involved, our collaborator has generated a series of block-deleted virus, each deleted with a block of the viral genome that's not essential for viral replication. And this clearly identified that the US29 to 34A block of the viral gene is necessary for the DMX-L1 degradation. So when this block of the gene is deleted, the DMX-L1 level go back. Then we want to understand which factor in this block is necessary. So we did a single gene block deleted virus screening. So this block, this single gene deleted series of virus encompassing every factor in this block. And this showed that the US33A gene, the virus, the protein of this virus, is responsible for DMX-L1 destruction. So when this gene is deleted, the DMX-1 level go back. So what is US33A? Surprisingly, it's a very small protein, only formed by 57 amino acids. And predicted by AlphaFold, we can see that it has a central alpha helix, but otherwise with unstructured region on either side. And its transcript analysis shows that this protein was expressed during the early infection as what we expected. So how this small protein can destroy the 3,000 amino acid DMX-L1? We did an interactome analysis. So each of 170 HCMV genes were tagged with a 14 amino acid residue called the V5, and they were expressed in the cells. So the interactors of this gene were trapped with the anti-V5 antibody bound to the base, and then they were analyzed for the interaction. However, this kind of reaction normally gives us like a list of the interactors up to 1,000 proteins, but only minority of them are true. And this was reviewed using based on the computationally machine learning strategy. And conveniently, the two highest confidence interactors of the US33A are RNF123 and UBAC1, which are the two components of the KPC complex. This is a key mediator of the protein destruction in a waste dump called the proteasome. So now we wanted to ask whether the RNF123 and UBAC1, the KPC complex, is necessary for DMX-L1 degradation. We used the CRISPR technology to knock out the RNF123 or UBAC1 or both in the cells, and then infected them with the wild type or US33A deleted virus. So let's see a picture of the DMX-L1 abundance in the unmodified cells first. So we can see that the wild type HCMV clearly destroys the DMX-L1. However, the US33 deleted virus could not. Then, things happened in the knockout cells, we found that either the RF123 single knockout or the UBAC1 single knockout or the double knockout, we found the wild-type HCMV could not degrade DMX L1, and it behaves similarly with the US33A deleted virus. And this shows that the US33A destroys DMX L1 using the KPC complex. So now we wanted to understand why. Based on its known function, the loss of the DMX L1 might lead to a less acidic lysosome. So we measured the lysosomal pH in the cells, so the control cells, and also the cells expressing the viral gene called the US33A. And we stained their lysosome using the lysotracker to show its lysosome pH. This is exactly what we found. The US33A cells got significantly lower lysotracker staining compared to the control cells. And this suggested that the DMX-L1's destruction by the US33A can lead to a less acidic lysosome. So what will happen if the lysosome is no longer acidic? I want to introduce you a process called autophagy. It is a very important recycling mechanism in the cells to remove or to reuse the damaged parts into the new part. So the autophagosomes can engulf the damaged or defunct cellular materials and then fused with an acidic lysosome. This acidic environment allows the proteins to be chopped up into the component amino acid or the membranes into the fatty acids, and then to allow them to be recycled and reused into the new part. So does DMX-L1 destruction impact autophagy? We measured this using a marker of the lysosome called the LC32, and this can be destroyed together with the autophagic cargo in this acidic environment, the autolysosome. So when the autophagy is inhibited, the LC32 level will be increased. And this is what we found. The LC32 level in the US33A expressing cells significantly increased compared to the control. And this suggested that the autophagy was inhibited by the DMX-L1 destruction. We want to look at that in more detail. Which stage of autophagy was inhibited? We measured this using the LC3, which is the autophagosome marker, and a marker of the lysosome, which is called the LAMP1. If the autophagic cargo degradation was inhibited, there will be more LC3 co-localized with the LAMP1, so all things are stored here. However, if the autophagosome lysosome fusion was inhibited, the LC32 will be separated more with the LAMP1. And here is the result. We found that in the US33 expressing cells, when the DMX-L1 was destroyed, the LC32 was largely co-localized with the LAMP1. So here, so the red plus the green bit to become this kind of like orange or yellow color. And this suggested that the DMX-L1 destruction inhibits the autophagic cargo degradation. So now, finally, we want to ask why virus want to do that. It's the DMX-L1 destruction, but the virus can benefit the virus itself. So what we do is we infect the cells using the wild-type HCMV and also the US33A deleted HCMV. And then we measured this virus associated with the cells and also the virus released from the cells. And here is what we found. The US33A deletion from the virus significantly reduced the proportion of the cell-associated virus. This suggested that the DMX-L1 destruction by the HCMV might enhance the viral intracellular survival, and this might enhance the cell-to-cell spread of the virus during the in vivo infection, so when the virus infects our body. So overall, in conclusion, what I found is that the US33A destroys DMX-L1 by rejecting the cellular KPC complex. And DMX-L1 destruction inhibits lysosomal acidification and the recycling of the autophagic cargo. And DMX-L1 destruction enables the increase the cell-associated HCMV and might help its intracellular survival and cell-to-cell spread. And targeting the DMX-L1 destruction by US33A is our future direction. So we will use this, and it might be a very good new antiviral therapies. So at the end, I would like to say many thanks to all the Weeks lab, especially my supervisor, Mike Weeks, and our alumnus, Alice Fletcher Etherington. And also I would like to say many thanks to the Rubinstein lab in Cambridge and which helps us a lot for our autophagy assay and our virus collaborator in Cardiff University, UK. And thank you very much for listening, and I'm happy to take any questions. Thank you.
Hi, very interesting talk. So I have two questions. So the first one, I was just wondering, so how essential is the DMX L1 degradation for the virus infection to work? So do you have any assays or experiments where you can artificially block DMX L1 degradation and still look at what happens to the viral infection?
So it means they in vivo or something? No,
so you're looking at the degradation of DMX L1,
yeah.
and that's correlated with, say, viral infection. But if, say, in a scenario where the viral infection is still there and DMX L1 is not degraded, does that make a difference? Is there any way to test it?
Well, thank you. That's a very good question. Yeah, when the virus infects the cells, the DMX is still there. But when we knock out the US33A, the factor that degrades DMX L1, and we found the virus that associated with the cells significantly reduced. So but the virus are still there. Yes, you're right. So it's a kind of one factor of that. But yeah, because the virus is very complex stuff, it also can regulate all the factors of the cells. So yeah, you're right. It's not the only single important things in the cells. It's never be. Yeah, but
what if you do it in an overexpression model of DMXL?
Overexpression of DMXL1 to...
Yeah, so would the virus still degrade all of the DMXL1?
Yeah, because it's when we infect the cells using the virus, we found that, when the US33 deleted, the DMXL1 level got back to 34-fold, which is far more than the normal expression level of the DMXL1. So we think the virus is very sufficient to degrade that. But you're correct, yeah. I think later maybe a overexpression of the DMX1 is worth to try. Thank you.
Great, thanks. So the other question, so when you delete the S33A or overexpress it in the virus, it probably has other effects than just degrading DMXL1, right? So for those experiments, you have a control to show that the phenotypes you see are DMXL1 degradation specific and not just a general effect of knocking out S33.
Knocking of the... Oh yeah, so it's we got another virus also to show this kind of thing. So we're not only using the US33A overexpressing cells for sure, and we're also using the adenovirus, and we saw exactly the same results shown here. And also we infected the cells with the wild type and also US33 deleted virus, and we saw another way around. So it's also can prove this, yeah, true. But here I just only showed the US33 expressing cells.
Right, great, thank you.
No worries.
Hello, good morning. So first, thank you very much for your very good presentation. So I just want to ask a very simple question. So based from your conclusions, what could be your future directions on your research? Thank you very much.
The future directions of this virus is so we will firstly prove it, whether this kind of DMX1 destruction can enhance the cell-to-cell spread of the virus. And we have a virus, actually, we already got that. It doesn't have the US33, and also it's only spread between the cells, so not released from the cells. And we'll do this one first. And also, it's in the future, if we can collaborate with some very clever or intelligent people doing the kind of drug design, and also doing the kind of AI and computational simulation of the disruption of this kind of protein-protein interaction, so we can futurely try to design kind of
thank you very much. Good luck and congratulations.
Thank you.
Thank you for the talks. So I have two questions. The first one is that, is DMX L1 expressed in every cells? And the second question is that, is this mechanism preserved in different type of cells, particularly like the immune cell and the brain cells?
Yeah, so the DMX1 is expressed in most of the cells. So like the HeLa cells also is not like, and the kidney cells and epithelial cells, they're all there, but their expression level was like relatively low in all the cells, including the cells we use, the fibroblast. And another thing is like, sorry, would you like to repeat your second question?
Yeah, is this mechanism preserved and conserved in different type of cells, particularly in male and brain cells?
We can try to figure this out. It's like because some of the neuron cells is permissive for the HCMV infections. We can try that in this kind of cells, but because of the the restricted cell type that HCMV can infect, so we only use the fibroblast. But in the future, that's a kind of direction to look at this kind of general thing. Thank you.
Thank you.
I have questions. So a lot of viruses, they use like endosomes to enter the cells, and also when they matures in a cell, they use also vesicles that are low in pH.
Yeah.
So CMV doesn't use any of those?
When CMV enters the cells, it doesn't use the endosomes to do that. Sorry.
So it doesn't need that?
Yeah, it can get into the nucleus very quickly, like within the 12 hours. It can also spread the kind of very early genes to start its replication. And way to get out, there is some evidence showing that exactly the autophagism, it can help the cells to egress from the cells. There are some literature already shown that. So when the cells getting out from the cell, it takes some bit of the autophagism. So that's why maybe when the DMX L1 destroyed, it can reduce the cell release as well. And also the thing is that normally HCMV, when it's getting out, it's using this kind of acidic environment to assemble its thing, but you can see it's like not assembled in one single endosome. When I show the infected cells, they're like the particles is kind of so messy in the cells. It's just distributed.
So in induced autophagy, so does it mean that the cell will die?
No, this autophagy is a kind of like recycling thing. So it can be associated with the cell death before, but actually autophagy is kind of like enhance the cells to live further. So they recycle the damaged part and the old parts to try to promote the cell's death instead of like inducing the cell death.
Okay. And one last question. So you use a drug to screen for the host target, right, that is bound by the viral protein. Yeah. So did you look at whether the drug actually binds to DMX L1 directly?
So actually, we didn't look at that. Yes, you're right. Yeah, true, because we use the drug as get into the with the DOMs such as the lysosome and the proteasome, but we didn't look at if it interacts.
So you don't know the mechanism of inhibition by the drug?
Inhibition of
the mechanism, how it does this inhibition.
How?
So the drug inhibits the infection, right?
We didn't have any drug to inhibit.
I thought you used a drug in order to get the mass spec.
Oh yeah, that's why it's like it's not. So the drug was applied before the infection, so it's just to get into the proteasome, and so it's MG132 and lupeptin. So it inhibits this kind of like proteasome and lysosome cell environment to allow the proteins not degraded that. So it might not affect the infection stuff, I think.
Oh, okay. All right. I think that's it then. Okay. Thank you, Han Qi.
Thank you.
Okay. The next speaker is Patrick Fuchs. He's a research fellow at the University of College London, and his research field is in medical physics. Yeah, you please start.
Thank you. Thank you for the kind introduction, Professor Locke. Thank you all for being here. It's a great honor to present to such a wonderful crowd. My research is on imaging the brain, specifically using magnetic resonance imaging and more specifically the magnetic susceptibility within the brain in this research. Before we get into the susceptibility, I will give an introduction on magnetic resonance imaging for those of you who are unfamiliar with how it works, because a lot of the intricacies of this research lie in the tool we use, the magnetic resonance imaging device. And also a lot of the things that are possible are because of the way the physics work. After that, I'll go on and give a bit of an introduction to magnetic susceptibility, although Sir Andre Geim yesterday has been very nice to demonstrate a diamagnetic susceptibility of water. So should be a bit more familiar to you today. Then quantitative susceptibility mapping is the method we use within MRI to image this. And finally, I will summarize some of our research specifically in looking at neurodegenerative diseases and functional contrast, as well as really diving into separating iron and myelin in the brain and looking at basically on a molecular level function in the brain. Because there's a lot we don't understand about the brain, and hopefully some of the methods I present today will, down the line, help us understand how our brain works, how memories work, and some of those things. But first, MRI scanners. I'm pretty sure most of you have seen these before, whether at a hospital or in a movie, but typically they feature a relatively large magnet. Most of the time in hospital settings, these will be super-cooled magnets, and they require a large room to shield the magnetic field from the rest of the hospital. These magnetic fields are very strong, often 60,000 times stronger than the Earth's magnetic field or more. And the big bore is essentially designed to apply a uniform magnetic field to the body inside of the bore. This magnetic field interacts with the tissue of the person inside the bore because hydrogen atoms have something that's called a spin in quantum mechanics. Now, for those of you familiar with quantum mechanics, you know it's not actually like a spinning top, but for this, it makes a lot of sense to present it as such an analogy. So you can imagine basically the proton spinning around its axis, even though that's not really what happens. This spinning motion means that a spinning charge introduces a moment, a magnetic moment in this case. This magnetic moment, therefore, is susceptible to an applied magnetic field. So in an MRI scanner, by applying a magnetic field, you can essentially align these spins with the applied magnetic field. Now, in MRI, we're not able to get our resolution down to single spins because that is a very, very small signal. But we're looking at ensembles of spins. So essentially in a single pixel, or because we're dealing with 3D volumes in a voxel, you have the net magnetic or net magnetization vector, which is the sum of all the ensemble spins. And the stronger the magnetic field, the more of these spins are aligned with that magnetic field, but most of them realistically point in random directions, unless you cool something down to very cold temperatures, which unfortunately most of our subjects would not like. So now that we have these spins aligned, how can we measure them? We do this by applying a radio frequency pulse that knocks them out of alignment. And because they're spinning around their axis, now that they've been knocked out of alignment with the magnetic field, they're spinning in the plane of that magnetic field, perpendicular to the applied magnetic field. And because this rotation is much larger than the precession that the magnetic moment had around its axis, we can measure it very well using radio frequency coils. And as we're measuring this, the spin slowly relaxes back into alignment with the magnetic field. In MRI, we repeat this process many, many times in a row because this relaxation typically takes in the order of milliseconds. And we slowly form an image from measuring these spins over time. So that brings us to susceptibility, magnetic susceptibility to be precise. What is it? Well, Sir Andre Geim yesterday explained that it means that an object interacts with a magnetic field. And even things that most of us would not consider to be susceptible to a magnet, because they're not attracted by a magnet, have this magnetic susceptibility. And that is why you could levitate a drop of water or even a frog in a strong enough magnetic field. Now, the magnetic fields we use in MRI are not strong enough to levitate the human body inside the bore, although that would certainly be a sight to see. But the same relationship between the magnetic field and the susceptibility holds, where diamagnetic tissue actually pushes the field out, and paramagnetic tissue attracts the magnetic field, as it were. So the paramagnetic tissue will focus the field in some regions, and diamagnetic field will decrease the field strength locally. And because we have this, so water, which is the most abundant molecule in our bodies, has a slightly diamagnetic susceptibility of about 9 parts per million. So watch the 10 to the minus 6 of this number line. And here I have some other elements that are relevant in the human body or within the context of the human body. For example, bone and red blood cells, which I'll get to later on. Now, the susceptibility of red blood cells changes between deoxygenated red blood cells and oxygenated red blood cells, where the deoxygenated ones do not carry oxygen anymore, and the oxygenated ones carry oxygen. But for most of the susceptibilities that I'm interested in the human brain, the difference between them and water is much smaller than this line. And typically, the contrast we're looking at is in the order of parts per billion. So 10 to 15 parts per billion, one order of magnitude smaller than the susceptibility of water compared to air. That is also what makes it so difficult to image, because the majority of the effect that the human body has on the magnetic field in MRI scanners is because of this shift from air to water. So a shift of almost 10 parts per million. This shift means that in the scanner, where we have this really nice uniform magnetic field, in fact, most of the field where we're measuring is not exactly uniform. Slightly diamagnetic tissue will mean that the field locally is decreasing, and paramagnetic tissue means it's locally increasing in strength, because it shapes by pushing out or attracting the field. Now, the field determines the speed at which this net magnetization moment is rotating through something that's called the Larmor frequency. And that means for a stronger magnetic field, we have a faster rotating spins, and for a lower magnetic field, it's slower. Now, in our measurements, we're not measuring just the magnitude of an image, but we're actually measuring a complex number, where the magnitude is determined by the strength of the signal, and the phase is determined by how far it has rotated in this plane. That means that in regions where the magnetic field is stronger, the net magnetic moment will have rotated further along in this plane and will have accumulated more phase. And where it's weaker, it will have accumulated less phase. And therefore, through the phase of our measurement, we have a measure of the local changes in magnetic field. Now, usually, when people show you MRI images, they don't show you the phase. And that is because there's not much to be seen in it. And the majority of this phase accumulation I'm showing here on the right is due to the shift of water to air. So the -9 ppm we saw before. And very faintly, if you look very closely, you can see some of the anatomical features that are on top of this shift. Now, how do we go from this measured phase to the actual underlying susceptibility? That's unfortunately an inverse problem, and it's an ill-posed inverse problem, because the model we use for this basically relates every single change in susceptibility to the change in field it induces as it being a tiny dipole or like a tiny antenna in tissue. That means every voxel contains the sum of all of these susceptibility sources, and every local susceptibility change induces a global change in field because it attracts the field towards it or it pushes the field out. And the field we're measuring is the sum of all of these contributions. So in reconstructing the local susceptibilities, we have to disentangle these local changes from the global sum of them. Unfortunately, because of the time I have for this, I won't go into much more detail on how this process goes exactly, because we also want to know about why we're interested in these susceptibilities. And the primary driver behind the interest in magnetic susceptibility was iron accumulation. It has been shown through biopsies or by iron staining brains that have been autopsied from people that have died from these diseases, that there is a lot of iron accumulated, especially in the deep brain regions of the red nucleus and the substantia nigra, where we don't see that in healthy individuals. Now, there's a lot still unknown about these diseases, so we don't really know whether the iron is causing the disease or whether the iron is caused by the disease. But imaging this will help us in understand the relationship between the ferritin or the bound iron that we see in the brain and some of these neurodegenerative diseases. Another thing that we see is loss of white matter with age or demyelination. And because myelin is diamagnetic, whereas iron is paramagnetic, or the ferritin is paramagnetic, we have a clear difference between those in how they interact with the susceptibility. Finally, what you can see here in this example image as well is there's deep brain vasculature that becomes very clearly visible in susceptibility maps because, as I said, the blood and blood oxygen content has a great impact on susceptibility. So it's a clear driver of contrast in our case. And lastly, calcifications in the brain occur often with age, and they also have a very distinct influence on the susceptibility. It's not just neurodegenerative diseases that we're interested in, it can also be epilepsy or other neurological disorders that can be of interest because they change the structure of the brain. But besides the structure, we also know that these neurological diseases change the function of the brain. And for that, we have a tool in MRI, which is called functional MRI. Functional MRI works by measuring the change in oxygen consumption. If your brain is active, as you're looking at these slides, your visual cortex is active. As you're fidgeting with your phone, your motor cortex is active. And that means that these regions of the brain use more oxygen than other regions of the brain. If a region of the brain starts using more oxygen, the hemodynamic response of the body starts supplying more oxygen. It does this by increasing the size of the veins and also by increasing the flow. This means that there's an oversupply of oxygen because the reaction supplies more oxygen than is in fact being used. And this oversupply of oxygen is in fact what drives functional MRI. The oxygen of the blood that flows out of these regions in turn increases because there's more supply than is being used. And as I mentioned, because deoxyhemoglobin or deoxygenated blood cells have a lower susceptibility, it decreases the susceptibility of blood. And this in turn increases the measured signal magnitude because it decreases the dephasing or decay of the signal. Now, conventional fMRI measures this at the change of signal magnitude, which is downstream from this effect. And the change in signal magnitude is also influenced by other factors in the human body. What we measure is the susceptibility. So, in fact, you could do functional susceptibility measuring using this. And we've done so recently with a new sequence where we've looked at a visual stimulus, which is in the visual cortex in the back of the mind. Here we see a conventional MRI, fMRI, based on the magnitude of the scan and our susceptibility activation on the right. And as you can see, the voxels that are activated here span a much larger region based on the magnitude, and that is because of the downstream effect of the signal amplitude increasing, whereas the susceptibility is very localized, and because of the crosshair you see on here, it's almost like the voxels are disappearing, but we see the activation in the same region of the brain, so we're fairly confident that it's based on the same functional activation, the same thinking that is being done. It's a more direct measurement of this. Unfortunately, there are some drawbacks of functional susceptibility over functional MRI, and one of those is that the susceptibility that we're measuring is very susceptible to physiological noise, and specifically breathing, because the lungs are typically filled with air. And as we've seen, there's a large difference in susceptibility between air and water. So as you're breathing, you're actually changing the field that we're measuring at the head level. And there's still a lot of work we have to do to make sure that this fit looks almost less noisy and more like the fit on the magnitude. Because a lot of the extra jumps you see in here can be attributed to, for example, breathing artifacts that we see. Now finally, to bring this all back to iron and myelin, with which I started. As I've shown you in these local magnetic susceptibilities, for those of you familiar with gray and white matter images or T1 weighted MRI images, you'd say you cannot distinguish between the white matter and the gray matter very well in this. And that is true because our susceptibility, or the susceptibility we could reconstruct, is the sum of all of these susceptibility sources, which means that if there's a region of the brain that features both diamagnetic as well as paramagnetic sources, they essentially cancel out. We're losing the contrast. Now, what I've done here is use a source separation technique, which allows me to decompose the image into diamagnetic and paramagnetic sources by including the magnitude at multiple time points. This unfortunately means that the measurement takes slightly longer, but it allows us to get a clear delineation of myelin here. We see white matter tracts, and a stronger contrast in, for example, the putamen in the brain. And these are some of the techniques that we're working with and are developing to image specifically the aging brain, the brain in general. Thank you for your attention. And I'd like to thank Professor Karl and Schmöhle's group for their help in putting together these slides, and the European Research Council for funding this research.
We now invite Prof Lok to moderate the Q&A session.
Okay, questions. Any questions? No? It's very specialized?
Wow. Either very clear or very difficult. Thank you. If there's any questions on MRI in general, that's also fine.
Hi. Yeah, my question's a bit about MRI in general. You mentioned a bit the drawbacks of the functional susceptibility, which is pretty cool. I've never seen that. Can you compare like the time scale versus conventional MRI? Can you like acquire the same TE?
That's a good question.
And SNR as well. I was wondering, like the signal change in fMRI is small, I know. So how does that compare for the functional susceptibility?
That's a great question. So for fMRI specifically, as you're measuring your magnitude, you're also getting your phase. So for the measurement, it takes the same amount of time. And we use the same type of sequences that are used in fMRI. For conventional MRI, typically people use so-called T1 weighted or spin echo sequences, which allow for removing the susceptibility artifacts which we're essentially using to image. So in conventional MRI, we require slightly different sequences that do not remove this artifact. But because fMRI is a technique that requires very rapid imaging, often less than 3 seconds per volume, to get that activation in a reasonable time resolution. Those sequences don't have this refocusing from a spin echo, and typically the sequences that are used already include the susceptibility artifacts that we're using in this case. Previously, people would often discard the phase information of an fMRI measurement. But as we see nowadays, it allows you to both correct for some artifacts as well as gives us this extra contrast to look at. So in terms of acquisition, it's the same amount of time. It's just that the magnitude is often, from a scanning vendor point of view, there's a bit more work on getting you a nice magnitude. So there's a bit more denoising methods that are applied. And it does not suffer from those susceptibility-related artifacts that we get from breathing, for example. So there's less physiological noise there.
Thank you.
Hello, I also have a, let's say, general question. You talked about these, let's say, local and global contributions to the susceptibility that are encoded in the face, and that you would have to solve an inverse problem that you don't have more time to talk about. And I was wondering whether you actually can solve it, and if yes, how you do it.
Yeah, so I said it's ill-post, which means we usually require some regularization to solve it. But it is relatively straightforward. It's a convolution operation, essentially with a dipole kernel. So it's a deconvolution operation you're doing. Most of the time, nowadays, people use nonlinear least squares type approaches to solve this.
Yeah.
I can talk for another 20 minutes about.
Yeah. And then is there something you have to, some kind of model you have to apply to make the deconvolution work?
Yeah. So the dipole kernel, the problem of the dipole is essentially that it has a lot of zeros because it goes from a positive to a negative value in both directions, and dividing by zeros doesn't really work. So typically, what we do is we just apply a regularization to reduce the effect that it's the attenuation that it has on noise, essentially. And the model we're applying is essentially the model we have for how susceptibility interacts with the field. So it's loosely derived from Maxwell equations.
And let's say computationally, I mean, can you do it in real time, or does it take?
So there are approaches which are essentially real time. The more advanced solvers, they can take a couple of minutes, depending on if you have a very high resolution scan, it could take upwards an hour if you have a slow computer, but it's very manageable.
Yeah. Okay. Thank you.
Hi. Thanks for your talk. I have two questions. The first one is, can your technique be used to identify the specific species of the paramagnetic and diamagnetic stuff in your brain?
That's a good question. And no, we really can't, because we're measuring basically, typically we're measuring the sum of all the contributions, and only recently have we been able to separate them into positive and negative sources by using the effect they have on the relaxation rate. Now, if you have a model for your tissue, say a multi-compartment model that describes how the different paramagnetic sources, say if you have multiple different paramagnetic sources, could interact with it separately, then that might be possible to decompose it. But because they, at least in the model we are currently using, whether it's a paramagnetic source of ferritin, of calcium, doesn't matter. They have the same effect, except that one would be stronger, but then you could say it has the same effect if the volume fraction is lower, right? If there's less of a stronger source, it's the same as if there's more from a weaker source.
Okay, so the second question is maybe a little bit off the topic. Is currently I think there are many new techniques used for imaging the brain. For example, the atomic magnetometer use quantum technique. Can you please comment on this kind of new technique?
Yeah, sure. So the big advantage we have in some sense is that it's relatively quick, and compared to, say, for example, PET imaging is nowadays becoming more and more ingrained for Alzheimer's, because it's possible to image the specific amyloid proteins that are, have been shown to be relevant for that disease. But PET is, as of yet, still much more expensive and more involved than an MRI scan. So this technique will allow us to soon scan a much larger patient cohort, perhaps over a longer time frame, to get more insight into how the disease progresses. Because we can put them in for 5 minutes and get these information, essentially.
You say
maybe can you discuss after this? So there are 2 questions online. The first one is, do you have any comments on integrating diffusion MRI and susceptibility imaging?
Yeah, so diffusion MRI typically requires a number of diffusion directions that are acquired. And depending on the way these are acquired, you could also use the phase from those acquisitions to generate susceptibility maps. They are different forms of contrast, and I would say they're complementary in some forms. People have looked at diffusion tensor imaging, which looks at the structure of basically, for example, the myelin sheath, which has a directionality that can be reconstructed. Susceptibility essentially is also a tensor, which means it has a direction. And if you acquire head acquisitions at different angles, so you rotate your head while you're in the scanner, which is not easy for most patients, you can also get a susceptibility tensor. And these have been compared and combined in the past to get a better image of the microstructure of the brain. So that has been done, yeah.
And the second question is, what quantitative metrics can be extracted from functional susceptibility imaging? For example, connectivity measures or ICA-based?
I think it's pretty much the same type of metrics that can be acquired from functional MRI. But because of the more specific nature of the connectivity, functional connectivity imaging, it remains to be seen whether those are robust enough to be used. So we're interested in, for example, also the resting state networks. In functional MRI, most of the time people look at the activation while someone is performing a specific task. But if someone's performing no task at all, then there are still regions of the brain that are correlated. And those are called resting state networks, which change over time, but also change with neurodegenerative diseases. So it's much easier to correlate those with disease progression.
Yeah. Okay. Okay, thank you, Patrick.
Thank you. Yeah.
Hi, thank you, Prof. Lok. Prof. Lok will now present the certificates of appreciation to our presenters. First, we have Dr. Patrick Fuchs. And next, we have Ms. Lee Han Chi. Okay, we've come to the end of this morning's Young Scientist presentations. Thank you to our Young Scientist and Chair of the session. Before we continue, I'd just like to remind the poster session participants to put up your posters at the campus center. We will now proceed to the plenaries. For the first plenary of the day, we have Prof. Jack Dongarra, who unfortunately is unable to join us today, and has sent us his pre-recorded talk on an overview of high-performance computing and the importance of AI, ML, and future requirements. We will now start the video. Please enjoy.
Hi, this is Jack Dongarra from the University of Tennessee. I'm the most recent recipient of the ACM Alan Turing Award, and I'd like to welcome you to the Global Young Scientist Summit. I'm going to talk about an overview of high-performance computing and the importance of machine learning and artificial intelligence and some of the future requirements. Hi, this is Jack Dongarra from the University of Tennessee. I'm the most recent recipient of the ACM Alan Turing Award, and I'd like to welcome you to the Global Young Scientist Summit. I'm going to talk about an overview of high-performance computing and the importance of machine learning